platform. So let us first discuss about the topic of speech, that is amelioration of apoptotic signaling induced with alcohol, a restorative attempt in the etiology of alcoholic neurogenic myopathy. Some disorders due to chronic alcohol abusing are alcohol withdrawal syndrome that poses significant clinical challenge arising from the spectrum of alcohol use disorder, and symptoms occur when someone stops using alcohol after a period of heavy drinking. Another one is Wernicke-Korsakoff syndrome, that is Wernicke's encephalopathy and Korsakoff psychosis. This is a neurological disorder caused by the lack of thiamine or vitamin B1, and symptoms include lack of energy, hypothermia, low blood pressure or coma, and lack of muscle coordination. Apart from that, alcoholic neuropathy, alcoholic cerebellar degeneration, alcoholic myopathy, and fetal alcohol syndrome are some important alcohol abuse disorders, and dementia and other cognitive deficits are one of them. If we see the societal impact of alcohol abuse, then what we see that high levels of alcohol consumption have also been associated with accidents, injuries, suicides, crime, domestic violence, rape, murder, and unsafe sex, as per British Medical Journal. So, the General Household Survey 1992 reported that the average weekly consumption of alcohol in UK was 15.9 units, that is 1 unit is equal to 1 glass of wine, or 1 measure of spirit, or zero point 0.5 pints of beer for men and 5.4 for women. In addition, 27% of men and 11% of women were drinking more than the recommended weekly sensible limits, that is 21 unit for men and 14 units for women. And heavy drinking is more likely among younger age groups as well. In a survey of 12,000 Welsh adults, Moore et al. Reported that 31.1% of drinkers aged 18 to 24 engaged in binge drinking, that is, drinking half the recommended weekly consumption of alcohol in a single session at least once in a week. And as per Australian guideline that recommended consuming no more than 10 standard drinks per week and no more than 4 drinks per day. And if taken more than this, then associated harm include poor academic performance, vomiting, passing out, accidents and injuries, damage to property, and interpersonal and sexual violence, etc. In 2019, the Ministry of Social Justice and Empowerment released the finding of a national survey commissioned to study the extent and pattern of substance abuse in India. And the survey showed that about 14.6% of the population uses alcohol, 2.8% uses cannabis products, 2.1% uses opioids, 1.08% uses sedatives, and 0.7% uses inhalants, with a small proportion of country's population using cocaine, amphetamine type stimulants or hallucinogens, etc., according to Hindustan Times News. And the survey also said that more than 57% or more than 57 million Indians are affected by harmful or dependent alcohol use and need help for alcohol addiction related problems. In extreme cases, the adverse outcomes of substance abuse are suicide and death, but in many cases, the aftermath of a boozeful evening is physical violence, the burden of which most often is borne by family member, especially wives, as per the Hindustan Times News. So here we objectify some objects here, that to investigate the alcohol challenged deterioration in the mitochondria and synaptosomes of adult zebrafish brain, and to investigate the protective role of vitamin B12 in alcohol-induced cerebellar degeneration and myopathy in adult zebrafish. So the molecular mechanism underlying beneath These alcohol-induced toxicity include formation of ROS as a byproduct of mitochondrial oxidative phosphorylation by the use of alcohol. So ethanol potentiate the formation of free radical like nitric oxide, peroxynitride, superoxide anion, and hydroxyl radical. And here electron, we can see that electron flow through electron transport complex in mitochondria, that is complex 1, 3rd, and 4th. Is coupled to transport location of proton to generate the proton motive force, and del P, which can drive ATP synthesis or dissipated by enhancing inner membrane proton leak through uncoupling. So overflow of these electron occurs with one electron reduction of oxygen at the level of complex 1 and complex 3rd, resulting in the formation of superoxide radical, which is converted to H2O2 by superoxide dismutase or reacts with nitric oxide to form peroxynitride. And here H2O2 or hydrogen peroxide is a potential source of highly reactive hydroxyl radical, and its degradation require glutathione peroxidase that is seized by ethanol. So ethanol suppresses antioxidant level in the mitochondria and potentiate free radical formation, and increases redox pressure through NADH and promotes formation of reactive oxygen species. This is the molecular mechanism what is going on at cellular level. Next one is we have used to cope up of this problem of alcohol abuse and alcohol toxicity at cellular level. We have used vitamin B12. So what it does actually, vitamin B12 is essential for body to function properly since it helps with processes like red blood cell formation, DNA synthesis, short-term memory, and more. And vitamin B12 is abundant in most animal products like meat and dairy. And if there is deficiency of vitamin B12, then it could lead to the accumulation of compound called homocysteine itself produce ROS, reactive substances, or yes, free radical. This buildup can lead to severe megaloblastic anemia and some other health condition. So symptoms of vitamin B12 or folate deficiency include extreme tiredness, a lack of energy, paresthesia, a sore and red tongue, mouth ulcers, muscle weakness, disturbed vision, and psychological problems which may include depression and confusing state of mind. So here is the scheme. What we did actually, we provided the zebrafish with some group, and we made groups in that group 1 served as control, group 2 served as alcohol, and we have provided 0.40% alcohol to that group of fishes, and vitamin B12 in as a positive control group. Then we have taken 4th group, that is zebrafish, that is provided with alcohol 0.40% with vitamin B12, and these are supplemented with diet, and dose of vitamin B12 was 5 milligram per kg. So this scheme include measurement of body weight and length, measurement of alcohol concentration in water after some days, every alcohol changes in dose of tank every day, and every time we have to change the doses of alcohol. And before the treatment, we performed behavioral study like novel tank, thigmotexics test, etc. And then some biochemical tests have been performed like superoxide dismutase, catalase, and expression of new and antibody. Some immunofluorescence study have been taken place, and biochemical and histological studies also have been carried out. Another one, if we come to the result of this study, then what we see that yeah, so after the experimental protocol, we come to results. Okay, so here are we used novel tank test for study anxiety-like behavior in zebrafish. So two most commonly employed assays for studying anxiety-like behavior in zebrafish are the novel tank diving test and the light dark test, that is that have been carried out in 2012 by Rosenberg. So this is quick to implement and sensitive to various pharmacological and environmental manipulation as well as drug screening in zebrafish. So the tank here is divided into some zones such as top, middle and bottom to facilitate behavioral analysis. And in the novel tank environment, zebrafish initially dive bottom to down to the bottom and gradually start exploring. And this gradual exploration reveal reduction in anxiety. So here we suggest that in normal or controlled fishes, they explore in all over the tank. And due to alcohol, see here, inhibited exploration have been inhibited and increase in freezing behavior have been noted. And there are so many erratic movements that have been noted in the and alcohol that is act as anxiolytic or anxiogenic drug actually. And vitamin B12, vitamin B12 here, alcohol plus vitamin B12 when we have given in this tank, then this will retain the condition near normal. So this videograph is based on assessment of anxiety related geotaxic test and representative videograph of zebrafish, that is represented by heat map based videograph displaying the specific pattern of exploratory behavior, and that will, can be quantified by test using concerning endpoints and graphical analysis, etc. Another one is thigmotaxic test. In that test, the zebrafish choose to near the edge and avoid the central open areas of thigmotaxic tank, which was performed following some previous method. The test was carried out in 37 by 27 by 18 centimeter tank, which is made of acrylic glass apparatus containing 5 centimeter of water. And zebrafish here were placed in the center of the instrument, and locomotory behavior were recorded with video capture software for 7 minutes and acquired the behavioral data using TalkStrack, a video tracking software. And the water in the test device was replaced after each recording to eliminate any signals dropped by the fish in their water. So here are the result. This suggests the anxiety-like behavior, and they fishes generally they move. They actually the anxious zebrafish exhibit thigmotaxis or wall hugging behavior. So wall hugging is more prominent in alcohol treated group than the control and vitamin B12 treated group. Another one is muscle biopsy that is performed here, and in this slide we can see that histopathological finding in the muscle zebrafish of different group by hematoxylin and eosine staining technique. And muscle of control group showing normal tissue architecture, however there is dilation in central vein and distortion of myocytes have been located in alcohol treated group. However, vitamin B12 is able to retain the condition near normal. Another one is the Purkinje fibers that is located in cerebellum, specifically cerebellum region, that is meant for the control and coordination in fishes and fine movement in their swimming and control the swimming pattern. So this photomicrograph shows the sagittal section of adult zebrafish brain cerebellum region, and the left panel represent some of the magnification with 100 mm magnification bar CCR region. Some region are GCL, these are granular cell layer and molecular layer, and then this one is Purkinje cell layer. So these three layers are prominent in control, and these are, however, alcohol treated group. It found that it is deteriorating, and vitamin B12 is in some extent they can retain the condition. So this is all about cerebellum, how it will affect the cells of Purkinje fiber, and vitamin B12 can retain the condition near normal. Another one is SOD and catalase assay, some biochemical tests that have been performed here. And see here, SOD and catalase are some of the enzyme. These are antioxidants enzyme, which are SOD is basically cellular antioxidant enzyme that protects cell from toxic effect of oxygen radical. And this create a first line of defense against ROS and RNS, and they catalyze the conversion of superoxide into oxygen and hydrogen peroxide. So this is depleted due to alcohol, and vitamin B12 can retain its values near normal. And another one important enzyme is catalase. Catalase is an enzyme that break down hydrogen peroxide into water and oxygen, and it is present in almost all aerobic organisms. They protect body from oxidative stress, and it is in the two form, that is Fe iron catalase and manganese catalase. So here catalase enzyme, that is affected by alcohol toxicity, and vitamin B12 here retain its level in some extent. However, not very much significant data have been found here, but these are a little bit effective. Another important biochemical enzyme, that is NAK transporter or sodium potassium pump. Sodium potassium transporter is one of the important enzyme that is located in cell membrane, and this pump is mainly implied in generating Na and K sodium potassium gradient across the plasma membrane, and in the determination of cytoplasmic sodium level it is important. It also functions as a platform for protein involved in different function ranging from signal transduction to the cytoskeleton. So NAK pump that have been discovered by Jens Christian, that is awarded a Nobel Prize in 1997, and these are nerve cell dependent pump that respond towards stimuli and transmit impulses. It helps in maintaining resting potential and regulates cell volume. So fluctuation in lipid composition significantly influence the activity of certain enzymes, or these enzymes are of course NAK transporter, and alcohol induced oxidative injuries can change the synaptosomal membrane lipid composition and change these membrane associated enzymes as well. So our results are in argument with the previous study and found that alcohol treatment altered the membrane bound enzyme like NAK transporter. So here NAK transporter, alcohol affect NAK transporter, and vitamin B12 is efficiently retain its activity near normal. Another one is immunohistopathology. The study of immunohistopathology or immunofluorescence study revealed the new N component in the brain cells. So immunofluorescence expression of new N, basically first of all I wish to discuss that what is new N. So new N or neuronal nuclei is a protein found in the nucleus and cytoplasm of most neuronal cells in the brain and other parts of nervous system, and it is used as a marker for post-mitotic neurons and also known as FOX3 gene product. So new N is used in diagnostic histopathology to help identify neuronal disease and tumors, and changes in the new N expression can indicate neuronal degeneration. So here the immunofluorescence expression of new N in the adult zebrafish brain is mainly observed in periventricular gray zone and tectum opticum, a region of the adult zebrafish, and less intense new N expression was noted in neuron of the PGZ of alcohol treated group compared to B12 or control group. Another one studied